All right, I'll give you one last quick bit of information before we get in here to meet on person. What we're going to do, we looked at a little development already. We went through fertilization, basically. All right, so grab some markers. So after fertilization, this is where development starts. That first cell, it's called the zygote. That is, the fertilized egg. It's called the zygote. What will happen next? Again, it depends on the species. Keep in mind, developmental patterns are different with each species and certainly with each lineage. That's kind of why we're looking at several different groups. So they'll have some similarities in early development, but they're not that closely related evolutionarily, so their development will change quite a bit. But what you'll find is, after fertilization, if you watch them, the first thing you'll notice, the first developmental process that's visible, is something called cleavage. So yes, we'll explore cleavage in a minute. Cleavage is really just described as rapid mitotic divisions. Now, of course, mitosis, again, it's occurring in your cells right now. Until you're dead, pretty much mitosis is occurring. What makes this particular type of mitosis unique is its very rapid division with little or no growth between divisions. So, when you look at that cell cycle, the G1, which is a normal growth and activity of the cell, S phase, which was DNA synthesis or replication, G2 kind of prep for mitosis or meiosis, and then the cell divides the nucleus and then goes through cytokinesis. Basically, the G1 phase is really short. Now, depending on the species you're looking at, after fertilization, that's when they'll begin cleavage. Now, in humans, we're a little slow. It might be 24 hours before you notice anything. But in the species we're looking at in lab, the sea urchins and echinoderms, they're fairly rapid. After fertilization, there's a bit of a delay. Depends a bit on temperature, that sort of thing, but approximately an hour, and then they'll begin to undergo cleavage. And the embryo will undergo cleavage about every 20 minutes. So we'll end up going from a single cell, the zygote. It will divide into 2 cells. That's cleavage. That's cleavage furrow. It's basically where cytokinesis occurs. 20 minutes later, it'll divide again. We'll have 4 cells. 20 minutes later, it gets a little messy after that. 8 cells and so forth. That's cleavage, rapid mitotic division. Now, it turns out, depending on which species you look at, they will all show different what they call cleavage patterns. So if you have an early embryo and it develops or undergoes cleavage about the same way, that's used evolutionarily to show that they may have some similarities genetically. On the other hand, if you have 2 species that are completely different, like a sea urchin and a chicken embryo, they're going to divide completely differently. So we're going to kind of have to look at cleavage in the 3 specimens we're looking at. I'll mention a little bit in placental mammals or humans as well, although I have no specimens. All right. So cleavage is rapid mitotic division. And really, if you look at the size of the embryo, or the conceptus at this point, the pre-embryo, remember, this thing's going to have a fertilization membrane around it, so you can visually see that on the microscope slides. That fertilization membrane is rigid. It doesn't expand. It's not very stretchy. As cleavage occurs, you will still see the embryo encased in that fertilization membrane. That indicates there isn't really any significant growth during cleavage. What's happening is you're going from one big cell to many smaller cells. So you're kind of increasing the relative amount of genetic material in cell membrane versus cytoplasm, going from one big cell to many smaller cells. Now, when you look at a variety of species, again, we're looking at sea urchins, amphibians, frogs, and we'll look at a chicken, an example of an amnio. The cleavage pattern is different. The first thing I'll mention is, depending on the amount of yolk in an egg, again, yolk is the material that contains the nutrients and the raw materials to support early development. Or all development in the case of a sea urchin. It turns out from species to species, there are different amounts of yolk in the egg. Sea urchins, for example, have relatively small amounts of yolk. I put little black dots in here to represent the yolk. Relatively small amount of yolk, and it's evenly distributed within the oocyte or egg cell. As we'll see, yolk tends to interfere with cytokinesis, with the cleavage furrows. Yolk is just, it's thick, it's viscous, it's relatively inert, it doesn't divide well. Because sea urchins have very little yolk, it doesn't have much of an impact. So when this cell divides, it just divides cleanly right through. These cells divide cleanly right through. If the egg or the cells divide completely, that type of cleavage, surprisingly enough, is called complete cleavage. Yes, it divides completely. There's another term for it. It'll be on that term sheet. You might see the term a holoblastic cleavage. It means the same thing. Complete cleavage is more descriptive. Holoblastic, I don't know, it will sound fancy. That's the older term. But they mean the cell divides completely. So that's one pattern. Now, as we'll see, this represents the echinoderm. If we look at a frog or an amphibian, frog eggs are larger, and they have a relatively large amount of yolk. So there's a big mass of yolk, and frog eggs will have some polarity. Because the yolk is heavy, it's dense, it sinks to the bottom of the egg, and it's always going to be at the bottom of the egg. In fact, this egg, when you look at it develop, the embryo in general, has a polarity. They refer to the end that has a heavy yolk sinking down into it. That's often called the vegetal pole of the embryo. This other end, which doesn't have any of the yolk, just the lighter cytoplasm, is the animal pole. You can see this when you look at the frog embryos. I think I even mentioned it when I go over the slides for lab. So frog eggs have a moderate amount of yolk, and it's not evenly distributed. So when a frog egg divides, it still has complete cleavage. So complete cleavage in echinoderms, complete cleavage in the frog embryos, but that yolk slows it down. So if you watch this cell dividing, it would very rapidly divide through the lighter yolk, but when that cleavage furrow gets to the yolky part of the egg, the vaginal pole, it slows down tremendously. It's still considered complete cleavage because eventually that cleavage furrow will make it to the part of the egg. But what happens as this egg divides, once, let's say we divide a second time here. Now, if you imagine you have 4 cells, 1, 2, 3, 4. The top 2 have the real light cytoplasm in it. The bottom 2 have that heavy yolk. As they continue to divide, these cells at the top will actually divide faster than the ones at the bottom, because that yolk tends to slow it down quite a bit. And so you'll be able to see this in the frog embryos. The animal pole will have lots of little small blastomeres. That's what they call the resulting cells. In division, just the general term is blastomere, they're just a cell. But the animal pole will have lots of smaller ones. The vaginal pole, because they're dividing more slowly, will have fewer, much bigger cells. Now that gets us to the last species we'll look at, or the last lineage. It's an amnio, the chicken, which follows a similar developmental pattern to a placental mammal, to a human. And while we don't look at the early chicken embryos in lab, because there's really honestly there's not that much to see, because there's a lot of differences and they don't photograph as well. But if we can imagine a chicken egg, and of course it's bigger than all these others, the chicken egg, or a zygote in this case after it's been fertilized, you've seen chicken eggs. They have a massive amount of yolk. So the yolk takes up almost the entire volume of the egg. In fact, again, the yolk's heavy, it'll sink to the bottom. There's a little kind of bump or dimple at the top of the yolk, which is always going to be at the top of the egg. You plop these things in water, it'll rotate, so the heavy yolk's down, and there's this little kind of cone at the top. That is called the cytoplasmic island. I'll put a PowerPoint on and show you a few of these pictures in a bit. I'll just splice it onto the end. So a little bit of light cytoplasm here, the rest of this is big, massive yolk. Well, what happens when the chicken egg divides? It has so much yolk that the first cleavage furrow will start right here in light cytoplasm. If it hits that yolk, it stops. So it starts to divide, imagine this direction, and it quits. Can't go through that yolk. Then the next division will be up here, then the next division here quits. Every time a cleavage furrow gets to that yolk, it stops. So what that means, you have this big mass here that basically it's metabolically almost inert. It doesn't do anything. Just this top clump here starts dividing more and more and more. So those cleavage furrows, because they don't go all the way through, guess what kind of cleavage they call this? Yes, exactly. It is incomplete cleavage, or the other term you might see is meroblastic cleavage. Too much yolk, the cleavage furrows can't go through the entire egg. So you've got two different cleavage patterns right here. You've got complete cleavage, which an example is in the echinoderms. Frogs have complete cleavage, although you can't see the yolk slowing down the cleavage furrows. Amniotes, chickens, mammals will do the same thing. We have incomplete cleavage. Insects are a different story. They also have incomplete cleavage, but we actually won't go over insects much. They have something called superficial cleavage, but it's incomplete, too much yolk. So that's one difference in cleavage patterns you'll see. You can have complete or incomplete cleavage. It's kind of a continuum. For our examples, the echinoderms, frog, and the chicken, complete cleavage, but it slows down a bit, so you get different rates of cleavage at the animal and vegetal pole. And then the chicken or amniote does not, it has incomplete cleavage. Those cleavage furrows cannot make it through that yolk. Now, the other thing that we really won't be able to see in lab very well, but it is an important characteristic. You kind of need a three-dimensional structure to notice this. But you can take all the animals and they can have complete or incomplete cleavage. Also, the pattern that the cells divide is different. For example, we've got one for echinoderms and the amphibians or frogs in our example. They have something called radial cleavage. All right, all radial cleavage is, let's see if we can imagine it. I'm going to have a cell that divides into two. So there's one, there's two. The cleavage furrows, as the cells divide, the cleavage furrows basically rotate 90 degrees. So the first one will, let's say, right here where my hand is. The next one will rotate, let's say, that 90 degrees. So you'll divide here. The next cleavage furrow might rotate another 90 degrees. So you kind of divide in the plane of the board. The next one, 90 degrees here, here, here. So in radial cleavage, the way the cells divide is always at 90-degree angles to the previous division. So what you'll end up with, I can't draw over the dark, but I'll give it a shot. You might have a cell here, a cell here. I'll make them different sizes a bit so I can distinguish. Let's say we're looking at 4 cells. Notice the cells are all lined up. There's a cleavage furrow occurred here, then 90 degrees here. If there were 8 cells, there'd one be 90 degrees in the plane of the board. So that cleavage furrow is rotating 90 degrees each time. The echinoderms and the frogs show this type of cleavage. We're not looking at any specimens that show a different type of cleavage. It's called spiral cleavage. In spiral cleavage, the cleavage furrows, they do not rotate a full 90 degrees. So you might have a cell that divides in half, so that's one. But then the cleavage furrow won't go a full 90 degrees. And really the way to tell, if you look at the embryo, terrible drawing, I'll give you some better diagrams on the PowerPoint. But notice this cell at the top, there's its cleavage furrow, it doesn't line up with the cleavage furrow on the layer below. So it's almost like you divide the cell this way, and then you just switch less than 90 degrees. Every time the cleavage plane rotates, it's not moving 90 degrees. So the cells don't line up, the cleavage furrows don't line up. That's spiral cleavage. That occurs, I think, in the mollusks, things like clams and snails. I think worms have this as well. So it's just a different pattern of cleavage. We won't look at any examples of that in lab. I really don't have one, but they're so poor, look at them with a microscope, you can't tell what you're looking at very well. And the last type I'll mention, again, we won't have examples of it, but this would be what you find in humans, placental mammals. We have something called rotational cleavage. What that means, imagine you're in the 2-cell stage. Here it is. 2 cells. A little dorky looking with our snowman. Those 2 cells. These cells will divide this direction, but then the other set of cells will divide 90 degrees off. So the cells are not dividing the same plane. So think of this, for this set of cells, the cleavage plane is this way, then it rotates 90 degrees, divide them differently. They call that rotational cleavage. And also keep in mind, in humans, cleavage is not synchronized. You know, a lot of times in my example, we have 1 cell, then divides into 2, then 4, then 8, then 16, then 32. Kind of holds for things like sea urchins a bit. Humans, not so much. The cells will not all synchronize and divide at the same time. So you can have odd numbers of cells. You know, you can have 7 and 11. They won't all be dividing at the same time. This one might divide, none of the others do. Gives you 5 cells. And a little later, that one might. So it'll be rotational, but it's also asynchronous division in placental mammals. Now, we're not looking at any labs that don't have any placental mammal embryos to speak of, but that's the different cleavage patterns. Now, I'll splice on the end a PowerPoint where I can look at better pictures than my cell snowman, but let me stick that on there. Oh, I do got to tell you. See, I was getting bored. This is our lab room for 153. All our specimens in the back. Got a bunch of sea urchins back there for the sex lab next week. But I got a little bored talking to no one, so they're here, a bunch of dummies today. See, I had someone to talk to. I think I've been in isolation too long. I don't know. But let me put on a PowerPoint. All right, so I just kind of went over, this is just some better pictures. What you've got here is a sea urchin and a frog. This is representing cleavage. So right here at the first cell, this is the fertilized egg or the zygote for the sea urchin and the frog. So you got a sea urchin here, frog here. And then if you go from your left to your right, this is just showing you some of the early sort of cleavage in these embryos. Sea urchin here, frog here. Now what you'll notice, these both have complete cleavage. So here's your first cleavage furrow. And I just call that the 2-cell stage. And then it divides again, 90-degree angle. So there's a cleavage furrow there and kind of one there at 90 degrees. It's hard to do that on this 2.5D. 4 cells, 8 cells. And the little gray dots they're showing you in inside these embryos, those are representing yolk. So they're trying to represent it evenly distributed, not a lot of it. And then we go down to the next one. And this is the frog embryo. Again, there's your zygote. But what I want you to note, there is your yolk. It's always going to sit at the bottom because it's denser and heavier. It sinks down to the bottom of the egg, so the egg will always be face down with the yolk at the bottom. That is the vegetal pole where the yolk sits. Animal pole is the opposite end. And in some of these embryos, that's where the whole embryo is going to develop. The yolk doesn't do much. But in the frogs, we do have complete cleavage. So they're showing you the first cleavage furrow here. And they're trying to represent it. Gets to that yolk, and it just moves slowly. It's still going to make it all the way through, as you can see on the next slide, but it goes pretty slow. So here's, again, 2-cell, 4-cell, and here's 8. Now, what they're trying to show you here, notice that these cells down here, the vegetal pole, they're actually kind of bigger than the ones at the animal pole, partly because cleavage is slowed down. It's going to take a little longer to get there. So those are 2 examples of complete cleavage, but it is showing you that if you have some additional yolk, it starts to slow things down. All right, now this slide, it's showing you 2 examples of incomplete cleavage. You can ignore the one down here. We're not going to mess with insects much. This is that superficial cleavage. They tend to divide around the outside sort of perimeter or cortex of the oocyte, and not much happens in the middle. But we're not going to look at any insects. We won't be tested on insects. They're interesting, but I don't have any specimens. But up here at the top, this is showing you a chicken egg. Now you're actually looking down at it. Imagine this were floating in a finger bowl and you're looking kind of down at the egg. So this little layer right here, see this kind of light colored bit here? That's the cytoplasmic island. That is the light cytoplasm. The whole bottom below, sort of the bottom of your field of view, is that massive amount of yolk. So what you're seeing, the first cleavage furrow, you can see it right here. It starts dividing through, but when it gets to that yolk, it stops. So it's not a complete cleavage. Hence, these guys have incomplete or meroblastic cleavage. So there's partial
division, not all the way through the egg. Then it goes 90 degrees, and you got a cleavage furrow going this way. The first one went this way, and so forth. But it's just dividing this big egg. It's just dividing at the top, just at the animal pole. Vegetal pole is not doing anything. It's just sitting there. There's way too much yolk to get the cleavage furrow to divide it. So it just basically sits there. So this is just cleavage at the top there at the cytoplasmic island. And again, you got different, 2-cell, 4-cell, 8-cell, and so forth. That's kind of the difference between complete and incomplete cleavage. Now, well, this isn't great. It's the best example I have showing you the difference between spiral cleavage, which is like in mollusks, and radial cleavage, which is pretty much everything we're looking at is radial cleavage. So in lab, don't put down spiral cleavage for anything. You'll be wrong because I don't have any good slides of it. But in radial cleavage, what they're showing you, you're looking down at basically 8 cells. So you got 4 on top, 4 on the bottom. They've made the top ones a little smaller so you can see through them. So the cleavage furrow for the first set of cells is right there. Kind of right here. I need a different color. I can't even see that. Right here. I'll wiggle it a little bit. Then it goes 90 degrees, goes right through here. And if you look at the bottom cell, bottom set of 4 cells, you can see those cleavage furrows, they line right up. Everything's rotating 90 degrees, so everything stays kind of stacked right on top of each other. The cleavage furrow goes right through both layers. Contrast that with over here. This is that spiral cleavage. So if you look, the top layer of cells, there's a cleavage furrow. There's a cleavage furrow. But you've rotated the cleavage plane not 90 degrees. It's right here. If you can imagine going through to the lower set of cells, there's one cleavage furrow. It's not 90 degrees from this one. It's only like, 45 on this diagram. And that's the second one. So the cells in the cleavage planes don't line up. It's like the bottom set divides and you rotate a little bit, but not 90 degrees, and the top set divides. So everything's a little kind of spirals as it goes up, basically. That is our example of spiral cleavage. And this is just the last one I'll show you. What you're seeing here, this is actually one of our sea urchin embryos. You can see the fertilization membrane, that nice, smooth, circular membrane around the whole embryo. And remember, that's fairly rigid. So this thing has not gotten any bigger than the zygote. But if you look at it, you got a cell here, you got a cell here, 3, 4, 5, 6, 7, 8. This might be, I can't count the cells, might be about 16. But that's all the result of cleavage. We've gone from one big cell to lots of smaller cells, but the whole organism isn't any bigger. It stays the same size. That's kind of how we describe cleavage. Let's see. So we'll be looking at these when we watch them develop. Let's see. And I think only other thing to mention with this stage right here at the end of cleavage, this mass of cells in here, it's called a morula. It's on your sheet of terms. I could try and write it with the mouse, but man, again, that makes it look like it looks like a 3-year-old writes it more. The reason it's called a morula, again, this is all old terms from descriptive embryology. A bunch of guys sitting around and gals looking at a microscope and say, "Hey, Jim, look at this thing. It looks like a mulberry." You've seen a mulberry or a raspberry, those compound fruits are all kind of bumpy. That's kind of what this thing reminded them of, so they refer to this thing as a morula. That's where cleavage ends. So it's divide. The whole thing stays the same size, so they have this solid ball of cells, and that's what they call a morula. It's a solid ball of cells. Maybe superficially looks a bit like a mulberry. After this stage, we're going to start to begin to see some growth in the embryo, so the whole thing will get bigger. At this point, cleavage is over. The cells are still dividing by mitosis at a very rapid rate, but it's no longer called cleavage because the embryo is starting to grow. I'll also mention, if this were a human embryo, at this stage in development, this is occurring in the uterine tubes as it's moving towards the uterus. It takes it several days to get there, but it stays the same size. And this fertilization membrane actually prevents implantation in the uterine tubes. This embryo cannot implant in the uterus as long as that fertilization membrane is in there. And that's what prevents things like tubal pregnancies, that sort of thing. Only if this thing gets removed will the embryo implant, which is going to happen in the next stage of development, something called a blastula or a blastocyst. The embryo will grow and it will shed this fertilization membrane. But we'll wrap it up there for cleavage. And like I said, this will probably be the last video lecture so we can get into class and I can talk faster and scribble more on the board. But that'll do it. Sound good? Here. Sounds great. Yeah, see. Too much isolation. I'll see you guys on Tuesday.